dimmed. No, let's see.
I have a question.
It's like it's
Okay, let's talk about cell cycle. How many of you like cell cycle?
Interesting.
It's interesting. Yeah, I like that. You guys think that's interesting. That's a good start. Hopefully growth doesn't want to have anymore. Then you just something.
seriously.
I know, you do. Okay, so let's talk about cell cycle. This will help you guys with your lab stuff too. By the way, all the pictures are uploaded for yesterday's lab people. All the groups, whatever pictures I could figure out and find, I have uploaded them. So group 3, by the way, back there. I gave you pictures from another section, so now you have actual cells to count and measure.
Thank you.
Would like them. I think you would like them. Maybe they won't, maybe they'll be like, made it difficult for us. They're so pretty. I found some really pretty. Okay, sorry. Like these. Are you guys excited to see your results tomorrow?
Sure.
That was not very convincing.
Oh, yeah.
Okay, so,
Yeah, I know.
what are we gonna talk about here? The biggest thing that we are going to be talking about is the cell cycle control system. And that's the one part that I really want you guys to focus on. We are going to talk again about how the microtubules and all that stuff, at the metaphase plate functions as well, but specifically the cell cycle control system is what I would really like you to pay a lot of attention to. And then we're going to briefly discuss the different phases of the cell cycle, with the G1 phase, the initial kind of growth phase, the synthesis phase where the DNA is replicated, and then there's another gap or G2 growth phase, and then your M phase where the mitosis is happening. So we're just going to focus on mitosis. We're not going to talk about meiosis for the purpose of this class for now, because we want to really focus on how cells control this whole process to begin with, and how do they decide when a cell needs to replicate and divide, and when not to do so, right? And when it is time to just die with control of cell number and cell size. So let's just kind of go over just an overview of cell cycle. So what we know from back in 9th grade biology, or even maybe before that, is that our cells, they grow and then they divide into 2 daughter cells, right, through the cell division process. And this whole thing is the circle of life, or cell cycle. So you have your original cell, the chromosomes segregate at some point, they divide into 2 identical daughter cells, and then they go through another round of growth and duplication and do this thing all over again. Well, there are some numbers for how long this whole process takes, depending on what type of organism you are. So if you're looking at an early fly embryo, this duration of cell cycle is 8 minutes. If you were looking at a prokaryote, a typical bacteria, that is like 20 minutes. That's it. And then early frog embryos, they typically take about 30 minutes. Our mammalian intestinal epithelial cells, so that's a place that's constantly rejuvenating, constantly replicating and dividing. You're getting rid of old damaged cells and you're getting new cells in. Our skin cells are another example of that are constantly, differentiating, growing. Blood cells, but blood cells have a much longer lifespan than this. But the mammalian intestinal epithelial cells, you are having on average replication process happening every 12 hours. And then if you were to grow some fibroblasts, again, some very prolific cells in culture, their life cycle is around 20 hours. Their cell cycle, one round of replication and division is about 20 hours. So typically, when we plate cells, we keep that in mind that about every 20 hours, you're doubling that cell population. So over a period of a week, you're going through about 3 cell cycles, right? 3 to 4 cell cycles. So eukaryotic cells, they go through cell cycle in 4 phases. So you have your initial growth and kind of hanging out stage, G1 phase. Cell is, completely able to do all the functions that it needs to perform. It's hanging out, it's doing what it needs to do. If it receives a signal that there is a need to divide and there is a need to make more of itself, it will shift into S phase, which is your DNA replication phase or DNA synthesis phase. In this phase, the cell is going to replicate its entire genome, right? All the genome is going to get replicated and checked and made sure in G2 phase. So the second growth and kind of gap phase is partially there because it's going to be where you're going to ensure that all your genome was copied correctly. If there are mistakes, they're going to be fixed. You again examine the environment and make sure that the environment still needs more of you and you still need to be dividing. And then the shortest phase, but again, one of the more important phases is the actual M or mitosis phase, where you are going to get the nuclear division in mitosis and then the cell division in cytokinesis so that you can have the 2 daughter cells creating from that. So what controls what happens when is a series of proteins and transcription factors? Yes.
What if it decides it should divide and it has 2 copies of chromosome?
Say it again. What does it do if it decides to?
So it's completed synthesis and it has 2 sets of DNA, but it just decides it shouldn't divide.
But there are no reason to divide, it will hang out. It will just hang out. There are cells in our body that are always ready to enter mitosis immediately if needed. Hepatocytes is our example of that. Hepatocytes many times are what we call multiploid cells. They are not just diploid. They've already gone through at least 1 round, sometimes 2 rounds of cell division, and they are ready to go in case an injury or toxin destroys liver cells and needs replication. At that point, they will trigger them into mitosis and they will all divide kind of magically at that exact same moment. It's kind of cool to watch when you do that. So if we were to take out, for example, hepatocytes from a mouse, right, a young mouse, we extract the hepatocytes, that acts as a trigger because you just like melted the liver and took them out, and you'll watch them all kind of synchronously divide at the same time. So yes, so what exactly happens during this process? Where were we? So you have several points at which you can, the cell can get signals to divide or to stop the process. The biggest one is in this G1 phase. You can have a setting where the cell no longer needs more of these type of cells, and they will get a signal that says, hey, there's enough of you, you are touching. One of those signals is simply through what we call contact inhibition. So it's called contact inhibition. So let's say, skin cell, simple model. I have my skin, it's completely sealed up. There is no need to make more skin cells. It's touching, this central cell has other cells touching it on all sides. So this is going to act as a signal for this cell that no longer needs to divide, right? So this is called contact inhibition. The cells are touching it on all sides. It will no longer go into cell cycle. And that is one way that they will exit cell cycle and enter a G0 phase at that point to maintain what's there and no longer enter cell cycle. If they have already entered cell cycle, the first part is at the transition between G1 to S phase. So they are going to grow and do work that they need to do until there is, again, a signal from the environment. It can be in the form of growth factors. It can be in the form of a sudden injury or toxin exposure that tells them, oh, there's some damage and we need to recover from it. And so what they will do, what that trigger will do is that it's going to shift the cell into S phase. And that's basically looking at, is the environment favorable, right? That's the thing. Does the environment require more cells of my kind? And is it favorable for cell division? Are there enough nutrients in the environment? Are there enough growth factors in the environment? And then it would move into the S phase. In the S phase, once it finishes S phase and goes into G2, it will not enter mitosis until it makes sure that all the DNA is replicated and it's done so with precision. If there are issues, it's going to stall and it's going to fix those issues and it's going to fix the damage and it's going to make sure that it can still be moved. It will also do another check of the environment and make sure there are still enough nutrients and space available for it to continue. And then once it enters mitosis, there's a third checkpoint. These are called checkpoints. So there are 3 checkpoints. One is G1 to S transition. The second is G2 to M transition. And the third one is during mitosis at the time of metaphase. It is going to check to make sure that all the chromosomes are properly aligned at the metaphase plate on the mitotic spindle and that they are going to be separated together the way they need to be separated equally. So those are your 3 important checkpoints. This first one is called G1 to S transition. The second one is G2 to M transition. And the third one is the mitotic checkpoint. So what exactly controls it? Well, there is this what we call mitosis promoting factor, essentially, MPF activity that we saw was present in the cytoplasm of cells that were ready to go into mitosis. So if you took cells that were that you knew were in cell cycle, that were going into mitosis, and you took that cytoplasm and you injected it into some oocytes that were not ready yet, right? So-called ready for transition yet. But if you injected that cytoplasm from M phase cell in here, they would trigger mitosis in these oocytes, even though they had no other reason to divide at this point. So there is some type of protein factor there, some kind of signal present in that cytoplasm that is driving these cells into M phase. Similarly, if you take cytoplasm from an interphase cell, a cell that's maybe in G0 phase or G1 that's no longer not in the mitotic process, those cells will not enter mitosis. They will just hang out and they will continue to do what they were doing before. So there have been a lot of research looking for these magical factors that cause this. And what they found were that there were these protein kinases. There's a whole group of them present that control which cell cycle, which portion of cell cycle a cell should go into. They will control the transition from G1 to S, maintain them in S phase, G2 to M, all the different cell cycles, they are controlled by a group of these proteins called cyclin-dependent kinases or CDKs. Each cell cycle stage has a different cyclin and a different CDK attached to it. And that's going to control, those combinations are going to control what's happening at any given time. So the cyclin-dependent protein kinases are activated by these cyclins. The cyclins, they're called cyclins because they cycle with cell cycles. Certain cyclins are only present under certain phases. Okay? So here is an example. You get, if you look at mitotic cyclin, the cyclin that is present for mitosis to trigger mitosis, which would be the mitosis promoting factor, right? It is going to peak during mitosis, then rapidly degrade during interphase. It will slowly reach the maximum, it will start to produce and then slowly reach its maximum. And then once the cell enters into the mitosis, it will again rapidly degrade and then go on the same cycle. This particular mitotic cyclin was found to be cyclin B. So there are, again, like there's cyclin A, B, D, E, one of them. Each one controls different part of the cell cycle. M phase cyclin controls triggering the cell into M phase. So what are these cycles? There they are, and you are required to know them. You have cyclin D. Cyclin D simply tells the cell it should go into cell cycle, okay? So cyclin D is produced when the cell shifts from G0 to G1, essentially, okay? So it will tell you that, it's saying that it is G1 cyclin, right? But it is not exclusive to G1. It's gonna be present all throughout cell cycle. So it starts in G1, it triggers the cell to go into G1, and then it continues all throughout cell cycle. If the cell is no longer going to divide, if it's a senescent cell, it's going to shift because there's no more cyclin D produced, okay? So cyclin D production triggers the cell into cell cycle. The second one is your cyclin E. Cyclin E is a cyclin at the checkpoint, and it is going to cause G1 to S transition. When this is produced, so remember, cyclin D is there, still there, right? So if we were to look for it, cyclin D is going to be always there during cell cycle. Okay, this one so this one, cyclin E is going to be just here, and then it will go down again. It will only be present in this period. So G1, this is E. It's going to be present only at that transition from G1 to S. It partners with CDK2. The next one up also partners with CDK2. And it is going to be specific for S phase, and that's cyclin A. Again, so E is going to, let me write this, S is going to be present during S phase only. So cyclin A is going to be for S phase. And then the last one that you have is cyclin B. Cyclin B is going to be for mitosis. Now, and that interacts with CDK1. So cyclin B and CDK1 work together to do the work they need to do. So cyclin B actually starts getting produced in G2. And it, like I said, slowly increases, and then when it reaches M phase, it then rapidly degrades, and that's your B. Okay, so cyclin B is going to be for mitosis. It also kind of is a trigger between G2 to M transition. So the maximum that you actually get is at that point of G2 to M transition. It rapidly degrades as the cell shifts into mitosis. Okay? Yes.
So it just stops and it just the cycle stops and
The cell cycle stops. No more cell cycle, no more cell cycling. It will just finish what it's finishing and then go into senescence.
okay.
Or G0 stage, yes.
Oh, I'm just searching.
But in the chart, should we pay attention that there's 3 forms of cycle D or just
This one,
or
you
just
know, for what you need to know for now is this. Yes, this will become a little bit important later, but that's a minor point. The biggest point is that you have these 4 cyclins, each one can be used to examine cell cycle. So let's say I was I want to see, I study saffron, right, in my research lab, and I look at its anti-cancer activity. And I wanted to see the role of saffron on cell cycle of my cancer cells. And I know that it's doing something there. I could look at the expression of each one of these cyclins and see exactly what point in cell cycle saffron is affecting. So let's say that when I check, I'm still getting cyclin D production. I'm still getting cyclin E production the same way in untreated and my treated. But when I go to cyclin A, my saffron treated cells no longer have cyclin A. That means they will not be able to go through S phase, that it's inhibiting them at S phase. Let's say it still has cyclin A the same way, but cyclin B is getting produced, it's never getting released, or it's not getting produced, then it's going to affect it at G2 to M transition or at G2 phase. So it's gonna help me see exactly where in cell cycle my drug affects the cell. Yes, yes, and yes. You had a question.
So my question was for cyclin B, so that is present for the G2 to M transition, but during the M, it just rapidly.
It rapidly degrades. That's actually what triggers the cell moving into M phase. So it goes up and up and up all throughout G2. And then it will, as long as it's there, the cell is not doing mitosis. And then it rapidly degrades as the cell shifts into mitotic phase. Yes.
All the cyclin does is like, it's signal to the protein, or a signal to the cell, like we need to shift into this phase or we need to stay in this phase, but it
Yes.
doesn't like assist in actually doing processes.
Sometimes, but the whole idea is that it's shifting, that it's a signaling molecule. It's helping the cell decide where to go, which in itself is a big function.
Gotcha.
But yes, it will interact with other binding proteins too, as you will see in a second. But this is the biggest function of these cyclin CDK complexes, is to shift the cell from one place to one part of the cell cycle to the other. So the activity of these CDKs are regulated by cyclins, right? And like I said, the active CDK right here is going to be bound to my cyclin, right? And it's only active if it's bound to the cyclin. When this cyclin is no longer needed, it is going to be ubiquitinated. So a chain of ubiquitin is added to my cyclin. This shifts it into destruction mode for the cyclin. The cyclin gets degraded by proteasomal complex, this barrel-like protein complex that destroys the cyclin. When it's destroyed, this inactivates the CDK, and now the CDK cannot function, right? So CDK obviously has to do something. Remember, what does CDK stand for? Yes, which means what type of activity does it have when it's activated? What's it gonna do? Well, it's an enzyme. What exactly? Who said that? For that. Yes, I think so. That's to do earlier, so you can. So yes, so cyclin-dependent kinases, that's what they do. They have kinase activity. They are going to phosphorylate. When things are phosphorylated, what does that typically do to proteins when they're phosphorylated? It directly impacts their activity level, right? By either activating or inactivating. So the whole way the cell is moving through cell cycle is through activation of specific cyclin-dependent kinases that are going to regulate which proteins are going to be active next. And you can imagine if you're going to be in S phase, what proteins you might be needing next. And then if you're going into M phase, what proteins you might be needing next. And those activities are going to be controlled by these kinases, which were activated only because that cyclin was present there. Okay, so there is those. So activity of other CDKs can actually have dual function, and cyclin B is in that category. So you have the mitotic CDKs, right? And you have the M cyclins, as we talked about, right? Which is the cyclin B. Now, they actually don't work all by themselves. They have another, they need helper proteins with them. There are two helper proteins that are working in conjunction with cyclin B and CDK1 that are going to together regulate whether the cell is in G2 or in M phase. So one of them is inhibitory kinase WEE1. If the inhibitory kinase binds, puts phosphates on this mitotic CDK, cyclin, one, or well, cyclin B here, CDK1, then even when the mitosis, the MPF factor or the cyclin B is bound to it, it's not going to be active. That's essentially all G2. Okay, so your M cyclin, which is cyclin B, binds to your CDK1, but it does not activate it initially because you have V1 phosphorylating CDK and keeping it inactive. Now, once the cell has gotten through all the checkpoints, it's checked and made sure the replication is complete, it's checked and make sure there's no mistake, you will get activation of this activating phosphatase CDC25. And CDC25 then will remove these phosphates and activate the mitotic CDK. And that's when the cell will actually go into mitosis. Okay? So here is that cycle. So cyclin B, as I told you, accumulates all throughout the GAP2 phase, all throughout the G2 phase, you're getting more and more and more and more cyclin B, but it is not going to initially activate your CDK. So cyclin B is bound to CDC2 or CDK1. They're both names for the same cyclin dependent kinase, but the cyclin dependent kinase is phosphorylated at all these places, so it can't function. It's dephosphorylated by your CDC25, which triggers mitosis. So what happens in that process is that you have your activated MPF, it's going to trigger mitosis, and the cyclin B is going to be rapidly degraded as a process as well. So that is basically how it works. And now you have your CDC2. It is no longer phosphorylated and it's just kind of hanging out. You'll get, again, in the next round of G2, you'll get cyclin B synthesis. It's going to start binding to CDC2 and getting phosphorylation with the V1 of all those little CDC2 parts so that it's no longer active. It's not going to be activated. And then it goes through those cycles again and again. So what triggers the cell into mitosis is dephosphorylation of the two phosphates put in by the V1 kinase and degradation of your cyclin B. Okay? Yes.
So the cyclin dependent kinases, are they the ones that are phosphorylating other proteins, or are they being phosphorylated?
It can be both ways. In this case, it's showing you how it is doing those both functions. In this case, it's showing you, so see, they themselves can be regulated by kinases too. Remember how we had last time or a couple of lectures ago, we talked about the cell signaling and we had that MAP kinase pathway where you had MAP kinase, and then MAP kinase. There were all kinases that also had kinase activities of their own. So they were phosphorylating the next thing in the process and getting phosphorylated themselves too to maintain the activity. So that's the same idea here, except here you have specific proteins. So there are two different proteins here. V1 is a kinase, but this kinase, the way it phosphorylates, it's actually inhibitory kinase. It's going to inhibit the function of CDK1. But here, your activated phosphatase is going to dephosphorylate, but that dephosphorylation is going to activate the kinase and then make it do the work it needs to do. So here you have it again, that you can have your CDK activated or inactivated with the helper proteins that can have inhibitory or activating effect. Here is an example of those. In this case, you have an active cyclin CDK complex. If you have an inhibitory protein bind to it, in this case, B27, it is going to inactivate the complex, even though you have both cyclin and the CDK both present together normally, which would lead to activation. So the cyclin CDK complex activation, deactivation is the first step of control. The second step of control is production of these binding proteins, right? These secondary proteins that can activate or inactivate our cyclin CDK complexes. Well, where do these come from? These come from specific transitional control proteins, checkpoint, what we call checkpoint proteins, that are going to control when things should happen. So this, like I said, there are several different places that you can have these happen. In your G1 to S transition, the C inhibitors can block entry into S phase, that P27 as an example of that. It will inhibit the cells into going into S phase, even though it was ready to go. So it had already produced those CDKs, right, and cyclins, and they were already activated, but then the environment was not favorable, let's say, right? Something happened here. And so the checkpoint protein will be like, and it will activate these inhibitory proteins, P27, for example, and inhibit that transition. Similarly, at the G2 to M phase, if there is DNA damage or the replication is not complete, P53 tumor suppressor protein is a very good checkpoint. It's a big checkpoint protein. And that can activate these inhibitory phosphatases or inhibitory V1 complex, for example, or CDC25 and block entry into mitosis, right? Depending on what it needs to do. Similarly, at the mitotic phase, remember when the metaphase plate, we looked at the chromosomes all lining up and that basket structure getting activated or inactivated? Well, that's done through APCC complex activation that will delay the exit from mitosis if chromosomes are not lined up properly. So each one of those checkpoints are controlled by a checkpoint or a series of checkpoint proteins that will then control, upregulate or downregulate other inhibitory proteins to start that process or stop that process. Okay, so here is an example, right? Basically, you have your different phases and different checkpoints. And at each one of the checkpoints, the checkpoint proteins are going to examine the cell, make sure it is still amenable to go continue cell cycle, proceed with S phase, or maybe not at this time, but maybe later. So it's just going to pause there. Or if the situation no longer is amenable at all, it will exit back into G0. If you have, instead of growth factors, suddenly now you have differentiation factors, it will completely exit this whole process and instead turn to terminal differentiation and become a differentiated cell. So there are several things that can happen at this G1 to S transition. That's a very important checkpoint. The most important one during cell cycle to continue or to stall or stop cell cycle. So here are some ways that you can control that at that G1 to S transition checkpoint. You need essentially continued production of growth factors in order to continue cell cycle, right? So those growth factors are what you would call mitogens. They are going to promote mitogenic activity or cell cycle activity. If you don't have those at the right mechanism, you're going to have inhibition of cell cycle. So let's say that you did have a mitogen, and a growth factor bind to a growth factor receptor, and it activated it, it would send in signal inward, right, through the mediator proteins, and it would activate the G1 CDK and the G1 to S transition CDK. Both of those are important for the cell to go into cell cycle. Once they're activated, they would affect cell cycle by translocating into the nucleus and activating genes related to synthesis of DNA, for example. So here is an example. Here is the retinoblastoma protein, RB protein. It is normally going to be bound to the transcriptional regulators that are needed for S phase genes, and it is going to keep them inactivated at that operator region. When you have the activated CDK for G1 to S transition, it's gonna phosphorylate the RB protein, which makes it inactive. It can no longer bind to the regulator. If it can no longer bind to the regulator, it's free to go, and the transcription regulator is free to work. And it can activate transcription of genes that would help the cell enter S phase. That's proteins talking to each other. So yes, so basically, if you get a mitogenic signal, you get activation of the related CDK cyclin complexes. Those activate into the nucleus to regulate transcription of genes related to the next phase of cell cycle. That's what you remember. Yes. Naomi, you had a question?
Oh, I was just gonna ask you about the mitogen again. So if the mitogen does not bind, I guess, to the receptor, then none of this happens.
If there's no mitogenic signal, none of this happened. Yes.
So like,
Sorry, I don't know why
like
it's like
contact
that.
inhibition and stuff like that, would like a mitogen be the thing that's?
Yes, so if there is mitogen happening, right, mitogen is binding, but there is contact inhibition trumps mitogenic signal. So even though the signal is coming in, there are other inhibitory signals getting produced because their cells are bound to each other. And there is no space. So the environmental stimuli would not be enough because there's no space to grow. Unless you're a cancer cell, then you can just pile on top of each other.
So I'm a little confused. So kinases can phosphorylate and be phosphorylated. How do they know which ones to do? Is it just because of the?
They don't know anything. It's just the signals that are coming in that are controlling whether they get phosphorylated or unphosphorylated.
So it's not like a difference in charge or anything like that. Okay.
No, what happens to them is regulated by what signals come in.
So they have no control.
They have no control themselves, no. Okay, so here is an example of what happens when cell was actively going through cell cycle and they got UV zapped, let's say, or they got exposure to something that was damaging, right? They had Roundup exposure, whatever, something toxic happened to them. Well, so here you have X-rays causing DNA damage. That's going to break DNA in some sort or cause mutations of some sort. That break is going to obviously trigger the DNA repair pathways, and it's going to activate protein kinases. One of, like I said, big checkpoint protein, both a G1 to S transition and G2 to M, and specifically in G2 to M is P53. P53 is one of those proteins that's made all the time. But if it's not needed, it's rapidly degraded as well. If damage happens, or if certain things happen, it stabilizes. Because that damage has happened, it will activate protein kinases that will stabilize this P53 protein by phosphorylating it. Once it is stabilized and phosphorylated, it can now translocate into the nucleus. It can bind to P53 response elements inside the genome and activate transcription of genes related to its function. Depending on what type of phosphorylations happen, it binds to different types of operator regions in the genome, activating different types of genes. One big gene that it activates is P21. P21 is a big inhibitory CDK inhibitor protein. It binds to activated CDKs, cyclin complexes in G1 to S and S phase, and inactivates them. So they can no longer continue cell cycle. It's gonna cause a stall, right? It's gonna arrest the cell wherever they were. They could have been in the middle of S phase and they will stop. They will not continue further. So P53, by getting activated, is going to transcribe inhibitory proteins that will lead to inhibition of cell cycle in this case. Yeah. Now, other things that can happen with your CDKs, once your synthesis CDK is activated, right? So you have the SCDK, it's active, it's gone in, it's activated genome. what it does inside is that it is going to activate your origins of replications. Remember those?
Mhm.
Right? So this SCDK, when it goes into the cell, into the nucleus, it specifically recognizes these origin recognition complexes. You know, it activates those and they are then able to come in and activate all the origins of replication, open them up and start the replication process. So the SCDK initiates replication by blocking by removing the block from there. Now, it doesn't just have to do this. Remember, it started the process, but it also has to make sure it doesn't continue to happen. So this activated SCDK will also block this replication from happening more than once. So once you have the replication started, right? So you have the DNA helicase coming in, it's unbinding the DNA, all that stuff is happening. It is going to the pre-replicated process. One of the things that it will do, the activated CDK will come and phosphorylate your CDC6 proteins and other proteins that are at the origin of replication. So they can no longer reactivate transcription. It's only going to do it once, right? So it started the process by translocating into the cell, and it's started the whole process of DNA replication. But once DNA replication starts, it phosphorylates those origin of replications so that they cannot be activated again. And so in each cell cycle, you only get the replication once. Make sense? So it's doing two things, right? It's starting the process and then preventing it from happening again and again, just once. Okay, so we are running out of time, so I'm gonna stop here and we will talk about the rest later. Everything except for the cell contact lecture. So in the next lecture, we'll finish up cell cycle, cell death, all that good stuff, and then we will be stopping at that point. So we will have cell cycle, apoptosis, we'll talk about the 5 big reasons cancers happen, the big hallmarks of cancer, and that's going to be finished for your exam. Okay. Actually, if you're bringing in your papers, yes, you can bring them here.
I just wanted to.
I think your answer for the past 3 times you talked was phosphorylation. I swear to God, I think you've said